Assalamu alaikum and Bismillah Rahman Rahim. Welcome back to the second part of this video where we were discussing about the changes during a muscular contraction. So now we will discuss what are the molecular bases of a muscular contraction, what type of changes happen during if we see them on the basis of molecules. So molecular mechanism is responsible for formation of ectomyosin complex that results in the muscular contraction. It includes three stages. First is excitation contraction coupling. The second is the role of troponin and tropomyosin, and the third one is sliding mechanism. We will be discussing all three stages in detail. First of all, what is excitation contraction coupling? It's very important to know this, that excitation contraction coupling is basically the process that occurs in between the excitation and contraction of a muscle. When the muscle is getting excited and when it's contraction, everything that is happening in between is excitation contraction coupling. So the process involves a series of activities which are responsible for the contraction of the excited muscles. Obviously, when the muscle gets a signal and it gets excited, it has to contract in response. And in between, everything happens in series, and all the series are interconnected to each other. So when a muscle is excited by the impulses passing through the motor nerve and neuromuscular junction, the action potential is generated in the muscle fiber. When the action potential is generated in the muscle fiber, it is because of the impulses. Now, the action potential spreads all over the sarcolemma, and it also moves into the muscle fiber through T tubules. We discussed about the T tubules in our previous lectures. So, the action potential spreads all over the sarcolemma and moves through the T tubules. T tubules are responsible for rapid spread of action potential into the muscle fiber. And when the action potential reaches the cisternae of L tubules, from T tubules, the action potential reaches to L tubules. These cisternae are excited. And when these cisternae are excited, the calcium which were stored inside the cisternae are released into the sarcoplasm. And the calciums from the sarcoplasm now move towards actin filaments to produce the contraction. So what is happening? It's when it started when the impulse reached the muscle fiber. It generated the action potential. The action potential spread it all over the sarcolemma. It moved to the T tubules. From T tubules, it reached to the L tubule. In the L tubule, the calciums were present, and the calcium then moved outside of the cisternae and went to the, to the actin filaments. Then the calcium ion forms the links or coupling material between the excitation and the contraction of the muscle. Hence, the calcium ions are said to be the basis of excitation contraction coupling. So now we know the basis of contraction coupling depends all on the calcium ions. So what happened in excitation contraction coupling? It's that the impulse generated, the impulse led the excitation of the muscle, the action spread all over the sarcolemma. It then spread it into T tubules. From T tubules, it went into the L tubules, and from L tubules, it released the calcium ions, and calcium ions then get attached to the actin filaments. Now we'll see what is the role of troponin and tropomyosin in all of this. Normally, the head of myosin molecules has a strong tendency to get attached with the active side of F-actin. And however, in the relaxed condition, the active side of F-actin is covered by tropomyosin. So therefore, the myosin head cannot combine with actin molecule. So what we are discussing over here is the normal condition in which the head of myosin uh, is having a strong tendency to get attached to the active side of F-actin. And we have discussed about it previously that actin, the head of myosin always get attached to the active side of F-actin. So, but if the condition is a relaxed condition, the active side of F-actin is covered by tropomyosin, and it won't expose itself. If it will expose itself, what will happen? It will get attached to the actin, and ultimately the contraction will happen. But in resting condition, obviously we do not need any type of contraction. So what happens is usually the active side of F-actin is covered by tropomyosin. Then the large number of calcium ions which are released from L tubules during the excitation of the muscle bind with troponin C. The loading of troponin C with the calcium ions produces some changes in the position of troponin molecule. So when they lead towards the loading of troponin C with calcium ions produces some changes in the position of troponin molecule. What happened? It in turn pulls the tropomyosin molecule away from effectin. And due to the movement of tropomyosin, the active site of effectin is uncovered and exposed. So the large number of calcium ions which were released from L tubules during the excitation of muscle, they have now to bind with troponin C. We discussed the structure of actin filament in which there was troponin C. And on troponin C, C is because the calcium ion gets attached to them. And when the troponin C gets loaded with calcium ions, they what do? They bring some changes in troponin molecule, and these changes pull tropomyosin away from effectin. And when the effectin is exposed, what happens is that the head of myosin gets immediately attached to the actin or effectin molecule. So this is the role of troponin and tropomyosin, and in short, it's that the calcium ions are responsible with the attachment to the troponin C, and troponin C exposes the head of the F-actin, and then the head of the myosin gets attached to the actin molecule. Now we have sliding mechanism and formation of ectomyosin complex. This is also known as sliding theory. The sliding theory explains how the actin filaments slide over the myosin filaments and from the ectomyosin complex during muscular contraction. This one is also known as Rasher theory or walk-along theory. So each cross bridge from the myosin filaments has got three components, namely a hinge, an arm, and a head. After binding to the active side of F-actin, the myosin head is tilted towards the arm of the actin filament and dragged all along it. So this tilting of head is known as power stroke. And after tilting, the head immediately breaks away from the active side and returns to the original position. So what is cross bridge? Cross bridge is when the myosin filaments who get attached to the actin filaments, the head of the head of the myosin gets tilted. And obviously, when it will tilt, this is the attachment, this is myosin, this is actin. And when it will tilt, it will move the actin along with itself. So this is power stroke. Now it combines with a new active site on the actin molecule, and the tilting movement occurs again. So one tilting movement, the other tilting movement, and the contraction continues. Thus, the head of the cross bridge bends back and forth and pulls the actin filament towards the center of the sarcomere. So that's how the sarcomere shortens and the contraction happens. In this way, all the actin filaments of both the ends of sarcomere are pulled. So the actin filaments of opposite sides overlap and form the ectomyosin complex. When this happens, the muscle will shorten further. The actin filaments from opposite ends of sarcomere approach to each other. The edge zone becomes narrow, and the two Z lines come closer with reduction in length of the sarcomere. However, the length of A band is not altered, but the length of I band is altered. Because the length of A band is not altered because with A band, the myosin filaments are attached and myofilaments are not moving, but the head of myosin is moving the actin filament upon it. So that's the phenomena behind the no altered change in A band length. And when the muscular contraction becomes where the actin filaments from opposite ends overlap, and the edge zone disappears. So overall, what type of changes occurs during muscular contraction? Length of all sarcomeres decreases as the Z lines come close to each other. Point 1. Point 2. Length of I band decreases since active filament from opposite sides overlap. Edge zone decreases or disappears. The length of A band remains the same. So where does the energy come from for all of this process? Energy for the movement of myosin head always comes from the power for power stroke is always obtained by the breakdown of adenosine triphosphate into adenosine diphosphate and an organic phosphate. So the head of myosin has a site for ATP, and actually the head itself can act as the enzyme ATPase and catalyze the breakdown of ATP. Even before the onset of contraction, and ATP molecules bind with the myosin head. So the energy for muscular contraction is always and always obtained from ATP, and ATP breakdowns itself, and it's broken down into ADP, adenosine diphosphate, and inorganic phosphate ion. When tropomyosin moves to the exposed active sites, the head is attached to the active site. Now the ATP cleaves into ADP and PI, which remains in head itself. The energy released during this process is utilized for contraction. When the head is all tilted, the ATP and phosphate ion are released and a new ATP molecule binds with the head. This process is repeated until the muscular contraction is completed. So what's happening that the tropomyosin moves the active sites and the head is attached to the active site. And once it's attached, the ATP is converted, is being broken down, and the energy is being released and utilized for contraction. And simply when the heads tilt, the broken down ATP that was broken down into ADP and PI are released, and a new ATP molecule binds with the head. So this process is repeated in order to provide continuous energy for the purpose of muscular contraction. Now, if it will keep on happening, how will the muscle relax? How will it will go into the repolarization? So the relaxation of muscle occurs when the calcium ions are pumped back into the L tubules. The calcium ions which were attached with the with the F actin molecules with the troponin C, they moved back. When the calcium ions enter the L tubules, calcium content in sarcoplasm decreases, leading to the release of calcium ions from troponin. It causes detachment of myosin from actin, followed by relaxation of the muscle. So immediately when the calcium is being brought back to its place into the L tubules, into the cisternae of L tubules, immediate relaxation of the muscle happens. And this detachment of myosin from actin obtains energy from breakdown of ATP. Thus, the chemical process of muscular relaxation is an active process, although the physical process is said to be passive. So now let's see what we have discussed so far. And after that, I will show you a very good animation on the excitation contraction coupling, the function of troponin and troponin C and tropomyosin, and the sliding theory as well. So in this chapter, we discussed about the resting membrane potential. The cell, when it's not involved in any kind of activity, it's in the resting state, it's in the polarized state, and the polarized state is at minus 90 millivolts, and the charges are positive outside and negative inside. And when the impulse is generated, and the impulse generated and the changes in the electrical charges happen, it leads towards the action potential. And a series of electrical changes that occur in the membrane potential are known as are of two phases, and they are known as depolarization and repolarization. So during depolarization, the muscle contracts, the cell inside becomes positive and outside becomes negative. And during repolarization, the inside of the cells becomes negative and outside of the cell becomes positive. So the depolarization occurs because of the influx of sodium ions, and repolarization occurs because of the efflux of potassium ions. Then we discussed about the excitation contraction coupling, and excitation contraction coupling is the whole process which is responsible for the from when the impulse is generated, the action potential started, which spread all over the sarcoplasm. Then action potential moved to T tubules, from T tubules to L tubules. The calcium was released, and then the calcium entered into the attached with the troponin C, and troponin C expose the head of F actin. And when it exposed the head of F actin, the site of F actin, the head of myosin filaments got attached with the actin filament, and it then pulled the actin filament, and that was known as power stroke. The tilting of the myosin head was known as power stroke. and then it resulted in the this was known as Rashid theory, walk along theory. And the energy for all this process is obtained from ATP. And the muscle relaxes when this ATP goes, when the calcium which exposed the head of effectin goes back to the L tubules. So now I will show you a video on cross bridge cycle.
Muscle contraction is at the basis of all skeletal movements. Skeletal muscles are composed of muscle fibers, which in turn are made of repetitive functional units called sarcomeres. Each sarcomere contains many parallel overlapping thin, actin, and thick myosin filaments. The muscle contracts when these filaments slide past each other, resulting in a shortening of the sarcomere and thus the muscle. This is known as the sliding filament theory. Cross-bridge cycling forms the molecular basis for this sliding movement. Muscle contraction is initiated when muscle fibers are stimulated by a nerve impulse and calcium ions are released. The troponin units on the actin myofilaments are bound by calcium ions. The binding displaces tropomyosin along the myofilaments, which in turn exposes the myosin binding sites. At this stage, the head of each myosin unit is bound to an ADP and a phosphate molecule remaining from the previous muscular contraction. The myosin heads release these phosphates and bind to the actin myofilaments via the newly exposed myosin binding sites. The two myofilaments glide past one another, propelled by a head-first movement of the myosin units, powered by the chemical energy stored in their heads. As the units move, they release the ADP molecules bound to their heads. The gliding motion is halted when ATP molecules bind to the myosin heads, thus severing the bonds between myosin and actin. The ATP molecules are now decomposed into ADP and phosphate, with the energy released by this reaction stored in the myosin heads, ready to be used in the next cycle of movement. The myosin heads resume their starting positions along the actin myofilament and can now begin a new sequence of actin binding. The presence of further calcium ions will trigger a new cycle.
In this video, we'll dive into your skeletal muscle cell, review the various structures that lie inside, and also the details of muscle contraction itself. For example, I'll discuss things like how these tiny proteins in our cells, myosin and actin, use calcium ions and ATP to ultimately shorten our muscles and generate contraction and force. Then, at the end of the video, I'll give you a couple of reasons why you should care about these details and why they're so cool by explaining things like rigor mortis, an interesting phenomenon where muscles contract after death. This is the second video in a multi-part series that details the process of skeletal muscle contraction. You ready? Let's do it. Welcome to PhysioFlip. As we saw in the last video, muscle contraction first begins with muscle excitement. This happens when a motor neuron releases acetylcholine, activating nicotinic acetylcholine receptors, allowing sodium to enter the muscle fiber and creating a positive graded potential. This increase in positivity then activates voltage-gated sodium channels, allowing even more sodium to enter and starting an action potential in the muscle itself. This muscle is now excited. That's great, but we don't have muscle contraction or force yet. The goal of this video is to explain how this excitation leads to muscle contraction in the process known as excitation-contraction coupling. First, the action potential is going to travel and spread along the membrane of this muscle fiber, just like it travels down the axon of a neuron. Muscle fibers, though, have these unique structures called transverse tubules, or T-tubules, which are basically folds in the cell membrane which allow the action potentials to travel down and deep within the cell. And here's why these T-tubules are so important. In our muscle cells, many of the structures involved in contraction are buried deep within the muscle fiber itself, away from the outer edge of the cell. For example, deep within the muscle cell, we have many of these cylindrical structures known as myofibrils. Myofibrils contain sarcomeres, the structures in skeletal muscle cells that are made up of myosin and actin, the proteins that are directly responsible for contraction and force in the cell. We'll talk a bit more about these later. But for now, just keep in mind that the action potential's job is to activate these sarcomeres in these myofibrils to start contraction. So how can these action potentials traveling along the surface of the cell activate these hidden structures that seem to be buried deep within the cell? That's where T-tubules come in. T-tubules are folds in the membrane, allowing the action potential traveling along the cell membrane to reach deep into the cell, activating many more of these sarcomeres in the myofibrils, as we'll see in a bit. But for now, the action potential is traveling down the T-tubule deep into the muscle cell. Let's see what happens next. The action potential first runs into this spider web-like structure known as the sarcoplasmic reticulum, or SR for short. The SR, which surrounds myofibrils, is basically a calcium storage center. So when you think of the SR, think of calcium. And this is important because calcium is heavily involved in muscle contraction. The SR is storing a very high concentration of calcium, and the action potential's job is to force the SR to release that calcium in order to cause contraction. And if we zoom in on this section of the cell membrane, I'll show you how the action potential causes this release of calcium. Zooming in, you can see that the SR is packed full of calcium ions. The reason the SR has such a high concentration of calcium is because of this protein transporter, known as the calcium ATPase. Like its name suggests, this protein transporter uses energy in the form of ATP to pump calcium from low concentration on the outside of the SR to higher concentration on the inside of the SR. So this pump is responsible for keeping the calcium concentration very high inside the SR. But to get our muscles to contract, we need to let that calcium out of the SR. So do the opposite of what this pump is doing. Here's how that happens. As the action potential travels down the membrane of the T-tubule, it eventually runs into a protein known as DHP. DHP is a voltage-gated receptor on the muscle fiber's membrane that is physically connected to another protein known as RYR. RYR, a calcium channel located on the SR membrane, is basically the gate or the door that can allow calcium to leave the SR to activate muscle contraction. When the action potential reaches DHP, DHP changes shape. DHP won't open, but it does change shape enough so that it can pull on RYR, causing RYR to open. For that reason, I like to think of DHP like this little guy that can pull the plug and open RYR once it's activated by that action potential. Once RYR does open, this then allows calcium to leave the SR based on its electrochemical gradient and floods the inside of the muscle cell with a lot of calcium. This calcium will then be used to activate the sarcomeres and generate force, which we'll discuss in a bit. But for now, let's recap the steps we have so far. First, the motor neuron released acetylcholine, activating nicotinic receptors on the surface of the muscle fiber. This then allowed sodium to enter the cell, creating a positive excitatory signal or graded potential. This increase in positivity then activated voltage-gated sodium channels, starting in action potential. Using the T-tubules to travel deep into the muscle cell, the action potential changed DHP shape, leading to RYR opening and releasing calcium from the SR into the sarcoplasm, or the cytoplasm of the muscle cell. Okay, we're getting close, but we still don't have contraction or force. We're almost there, I promise. To see how force is generated due to this calcium being released, let's zoom in onto a small section of this sarcomere. In this sarcomere, we can see a variety of different proteins. Here we have the thick filament, composed mainly of the protein myosin, and the thin filament, composed mainly of the protein actin. Along the thin filament, we also have a couple other proteins, including tropomyosin and troponin. These four proteins together will help control if or how much force this muscle fiber sarcomere can generate. To generate force in our muscles, myosin has to bind to actin. It has to grab it, pull on it, and shorten the sarcomere. However, myosin normally cannot bind to actin because the protein tropomyosin is blocking access to actin. You can see the binding sites for myosin here on actin being blocked by tropomyosin. That's why I kind of like to think of myosin and actin like two teenagers who want to date. Myosin just wants to go out with actin, but there are these parents, tropomyosin and troponin, who are blocking that, preventing it from happening. But for this muscle to contract, we need myosin to bind to and pull on actin. So let's go through the steps that allow this myosin to bind to actin and eventually pull on it to generate force. First, myosin will bind ATP and break it down or hydrolyze it into ADP and an inorganic phosphate molecule. The energy released from this breakdown of ATP is then used to push the myosin back into position to an energetic state so it's ready to bind to actin. But tropomyosin is still in the way. The parents are still blocking myosin from dating actin. Well, that's where calcium comes in. Calcium can bind to troponin, activating both troponin and tropomyosin, which causes tropomyosin to change shape and expose the binding sites for myosin. That's why releasing calcium from the SR was so important. Without calcium, myosin could never bind to actin and would never generate force. Now with tropomyosin out of the way, myosin will now bind to actin, and by releasing the phosphate, generates the power stroke, which is just a fancy name for saying that myosin actually pulls on actin and generates force. At the end of the power stroke, the ADP molecule is also released, and the myosin will stay attached to actin in a state known as rigor. If we want to generate even more force, we need to just repeat this cycle. That will happen when a new ATP molecule binds to myosin, releasing the myosin from the actin. The ATP is then broken down just like we saw before, with the energy being used to push back the myosin and get it back into the ready and more energized state. If calcium is still present, myosin will bind to actin again and repeat the process to generate even more force. Let's quickly recap the steps of this process, often called the cross-bridge cycle. First, myosin breaks down or hydrolyzes an ATP molecule, using that energy to push the myosin back into a more energetic state and get it ready to bind to actin. Myosin will then bind to actin if calcium has been released from the sarcoplasmic reticulum. Myosin then releases the phosphate group, pulls on the thin filament, and slides it towards the center of the sarcomere, going through the power stroke and generating force. Next, myosin releases the ADP molecule, forming an even tighter bond with actin and entering a state known as rigor, in which the myosin has pulled on actin, is generating force, but is still stuck to the thin filament. To release myosin, either to help the muscle relax or to get myosin ready to start another cycle to generate even more force, myosin will bind ATP, releasing actin, and allowing us to start this process all over again. That's a lot of steps, right? But here's the result. Myosin will pull on actin and shorten the sarcomere to generate force. By the way, you might notice that neither actin or myosin filaments are actually getting shorter. All that happens is that actin slides over myosin, being pulled towards the center of the sarcomere, shortening the sarcomere to generate force. That's why this process is often called the sliding filament theory of muscle contraction, since the thick and thin filaments seem to slide over one another. So we've made this muscle contract, but we don't want our muscles to be contracting all the time. So how can we get it to relax? Well, we just turn everything off. Everything that we just did, we just stop doing it. The motor neuron has to stop firing action potentials, to stop releasing acetylcholine. And without that acetylcholine, the muscle won't get excited and won't start action potentials. But without these action potentials traveling along the cell membrane and down the T-tubules, DHP cannot change shape, RYR will not open, and calcium will stay in its cage inside the SR. In this case, the only thing really happening is the calcium ATPase pump is using ATP to pump calcium back into the SR, preventing myosin from binding actin, and therefore helping our muscles relax. All right, I know what you're thinking. Who cares? Seems like a lot of details, but I'm going to run through 2 reasons why you should care about this stuff a lot. Reason number 1, it's just cool. All the details we learned in this video are actually happening inside your skeletal muscles every single time they contract. In fact, I can prove to you that this is how your cells really work. Remember the sarcoplasmic reticulum, that calcium storage center? Here's an actual image taken by a high-resolution microscope showing the sarcoplasmic reticulum surrounding your myofibrils. This is actually what it looks like inside your skeletal muscle cells right now. And remember that sarcomere? You can see that under the microscope too. The striations or horizontal lines here are the filaments in your sarcomeres. You can also clearly make out the T-tubules running along either side and even parts of the sarcoplasmic reticulum as well. So everything we learned in this video is actually happening every single time your eyes blink. Every time you get up, every time you use your diaphragm to breathe. It's because of DHP, RYR, calcium, and all these players we learned about in this video. Pretty awesome. But you might not be convinced. Let me try this. Reason number 2 to care. Rigor mortis. Rigor mortis is an interesting phenomenon in which skeletal muscle cells contract or experience rigor after death or post-mortem and can actually stay contracted for as long as a few days. But why would muscle cells contract after death? Let's explain why this happens using the material we learned. We learned that you definitely need to release calcium from the SR to cause muscle contraction. Post-mortem though, motor neurons cannot fire action potentials, which means acetylcholine can't be released, and action potentials cannot be started in the muscle. But we learned that these action potentials were necessary to cause calcium to be released. So how would muscles contract after death without action potentials happening? Weird. Well, it turns out that after death, the membrane of the SR begins to break down and spontaneously releases calcium without any action potential required, which allows for force to be generated. Additionally, in rigor mortis, not only do the muscles contract or experience rigor, but they also stay in that state for many hours or even days. Why is that? The reason is because after death, our body also stops being able to make ATP, and the cells eventually run out of this valuable energy source. But remember, we learned in this video that ATP was necessary to get myosin to unbind and release actin to cause the muscle to relax. So, without ATP, the myosin will be unable to release actin, keeping the muscles contracted or in what we call the rigor state. So here's what happens in rigor mortis. Myosin can still bind to actin. Since calcium is being released from the SR, since the SR membrane is breaking down over time, myosin can then go through the steps we learned, pulling on actin and generating force, and entering the normal state of rigor. But after death, when the cells eventually run out of ATP, this next step is no longer possible. Myosin can no longer unbind actin. So when the cells run out of ATP, the myosin molecules will be stuck in this rigor state, in the contracted state, leading to rigor mortis. And that's rigor mortis, a unique example of skeletal muscle contraction without the need for neurons, acetylcholine, or even action potentials.
So that was all about excitation contraction coupling and what we have studied so far in this chapter and in the other chapters about the skeletal muscle. This was just amazing video, and I hope you just like this animation a lot and have got your concepts more clearer with this animation. So until next time, take care of yourselves. Allah Hafiz.